Hi guys, I'm back with another video of 101 Class Survival Guide where we help you get through difficult topics of various subjects. The topic I'll be discussing today is the Maxam Gilbert sequencing, and I've covered this topic in a very easy way along with some tips for you to help interpret the results correctly. The Maxam Gilbert sequencing method, also known as the chemical degradation sequencing, was developed in 1977 by Alan Maxam and Walter Gilbert. It is a chemical-based sequencing method, and it was published 2 years after the Sanger sequencing method. Only 24 nucleotides were sequenced when this method was developed. The principle of this method is that DNA is heat in nature, radiolabeled, modified and cleaved using chemicals and run on polyacrylamide gel electrophoresis. After that, it is observed using autoradiography because it is phosphorus 32 labeled. The base modifying and cleaving chemicals have some properties and conditions which include that they are base specific, they are added separately, samples cannot be mixed, and each sample is loaded into an individual well in the polyacrylamide gel electrophoresis chambers. So discussing the components, the first one is the single-stranded DNA which is made by the heat denaturation. The second one is the phosphatase enzyme which removes the 5' N phosphate. The kinase which helps add the P32 to the 5' end, and then there is the P32 itself. Then come the modifying chemicals which are the hydrazine and the DMS. The purines will react with the dimethyl sulfate which is the DMS, and the pyrimidines will react with the hydrazine in such a way as to break the glycosidic bond between the ribose sugar and the base, displacing the base. The chemical cleaving agent is the piperidine. The piperidine will then catalyze the phosphodiester bond cleavage where the bond has been displaced. The dimethyl sulfate or the hydrazine will attack the purines or the pyrimidine rings, whereas the piperidine will cleave the phosphate bond at the 3' carbon. So this is how the cleavage occurs. So the modifying agents include the ones that modify the pyrimidines and the purines. The pyrimidines include the cytosine and thymine, and the purines include guanine and adenine. Hydrazine and piperidine in sodium chloride will only cleave the cytosine nucleotides, whereas the hydrazine and piperidine will cleave both thymine and cytosine nucleotides. It has to be noted that piperidine will react with all of these. So the next one is the dimethyl sulfate and the piperidine alone, which will cleave the guanine. And the DMS along with the formic acid will cleave both the guanine and the adenine. The steps are pretty simple. First comes the DNA extraction and then the heat denaturation from which we obtain the single-stranded DNA. Phosphatase addition helps remove the 5' N phosphate, and the kinase helps add P32 which is radiolabeled to the 5' end from which the phosphate had been removed. And then we divide it into 4 parts, and each has a base modifying agent added to it along with the piperidine. The base modifying agents and the piperidine help cleave as mentioned in the previous slides, and after that we incubate it for some time. Page is run, which is polyacrylamide gel electrophoresis, and final step is the autoradiography, which helps us see where the DNA samples are. Okay, so now we're at the most important part of the presentation, which is the Maxam Gilbert sequencing, base calling, and along with its tips. So first of all, let me tell you what the base calling is. So it is an order of the bases from 5' to 3' after running the samples of the different lengths on the gel. So, how do we know which base comes first and if both guanine and adenine have been cleaved? Or both the thymine and cytosine have been cleaved. So, which base is present? The simple trick is to take the common in the both of the doubles, and if there is no common base, then the other one is the answer if a band is present for the other one. For example, in this one, the guanine and adenine have been cleaved in this sample, but there is no band here for the guanine. So it means that the answer here is adenine. Here we can see that the answer here is adenine. However, in the next one, there is no band for both of them. So it means that there is no band or there is no base or nucleotide for this one. And in the next one, we can see here that there are bands for the both. So here we take the common one, the answer. The common one here is the guanine. So the answer here is the guanine. See, it's pretty simple. So the conditions for the base calling include the guanine when the guanine and the guanine plus adenine both have a band, and the adenine when only guanine plus adenine gives a band, the cytosine when the cytosine and cytosine plus thymine gives a band, and only the thymine when the thymine plus cytosine gives a band. So the important interpretations include that the first nucleotide has the phosphate 32 here, and that the farthest sample is the smallest. This one here is the smallest one, which includes only the cytosine. Each nucleotide is added to the previous order. Here we can see that this one here is added to the sample as we go from the 5' to 3', as we go from the bottom to the top. The reading is just like the one in the Sanger sequencing gels. So this was pretty simple, and I think that you guys must have understood this. Thank you for watching. Please subscribe for more, and if you want me to cover a relevant topic or you have any query about this video, please feel free to comment below. Thank you very much.